Functions of ecosystem. Primary one is the nutrient cycling. We have already seen that while energy does not recycle through an ecosystem, nutrients do. Since the inorganic elements move through both the biological and geological world, we call them biogeochemical cycles. Of the 30 to 40 elements necessary to life, 6 rank as the most important: carbon, nitrogen, oxygen, nitrogen, sulfur, and phosphorus. These nutrients move from non-living to the living and back to the non-living again in a cyclic manner. Each element has its own unique cycle, but all the cycles do have some things in common. Reservoirs are those parts of the cycle where the chemical is held in large quantities for long periods of time. In exchange pools, the chemical is held for only a short time. The length of time of a chemical is held in an exchange pool or a reservoir is known as residence time. The length of time of a chemical is held in an exchange pool or a reservoir is known as residence time. The oceans are the reservoir for water, while a cloud is an exchange pool. Water may reside in an ocean for thousands of years, but in a cloud for a few days only. The biotic community includes all living organisms. This community may serve as an exchange pool and serve to move chemicals from one stage of the cycle to another. For example, the trees of the tropical rainforest bring water up from the forest floor to be evaporated into the atmosphere. The energy for most of the transportation of chemicals from one place to another is provided either by the sun or by the heat exchanged from the mantle and core of the earth. The biogeochemical cycles are of two basic types: gaseous cycles such as nitrogen and carbon, and reservoir is the atmosphere or hydrosphere, ocean. Then sedimentary cycle, which is very sedimentary, such as phosphorus cycle. The reservoir is a lithosphere. The nutrients are first taken up by the producers, bound in the organic compounds, carbohydrates, proteins, lipids, etc., and move along the food chain to heterotrophic level and ultimately from all trophic levels with the detritus to the decomposers. The decomposers break down the complex organic compounds and release the nutrients back to the soil, from where they are again taken up by the plants, thus making the cycle complete. These biogeochemical cycles give an insight in how human activities lead to eutrophication in water bodies and global climate change. Then ecosystem processes include the processes by which transfer of energy and nutrients take place in both between biotic and abiotic components of ecosystem. The storage and fluxes are two main parts of ecosystem process. Storage means accumulation of chemical compounds in the ecosystem components, and flux indicates the conversion or movement of stored chemicals from one component to another. Food chains and food webs. These processes mainly include synthesis of food by the producers, transfer of energy and nutrients contained in food through different levels of food chains, and food webs, returning back of nutrients to the soil by decomposers. Various ecosystem processes and their controlling factors are briefly described, like photosynthesis. The primary function of photosynthesis is to convert solar energy into chemical energy mediated by the plants. The plants' living systems are powered by this process. The primary function of photosynthesis is to convert solar energy into chemical energy mediated by the plants. Plants' living systems are powered by this process. By the presence of sunlight, green plants take carbon, hydrogen, and oxygen from carbon dioxide and water and then combine them into glucose molecules and O2 as a byproduct. In the process of photosynthesis, carbon dioxide is fixed or reduced to carbohydrates, glucose, and water is split in the presence of light to release O2 molecules. This is to mention that here that O2 released comes from the water molecule and not from CO2. So the O2 is from O2 is coming from water, not from CO2. The overall photosynthesis process can be written as carbon dioxide plus water plus light gives carbohydrate plus oxygen, and can be represented by the following chemical equation: chlorophyll with the help of chlorophyll. See 6 CO2 plus 12 H2O gives C6H12O6 plus 6 H2O plus CO2. 6O2. Sunlight energy is stored in the bonds of glucose molecule. This energy is transferred into animals and humans when they consume plants. Further, O2 released in the process is used by plants and animals during respiration. Thus, photosynthesis and respiration go hand in hand. The process of respiration breaks apart the glucose molecules, and the released energy is then used for all metabolic processes. Photosynthesis at all photosynthesis reactions take place inside chloroplasts, small organelles present in mesophyll cells. In leaves and stem, chloroplasts are green due to the presence of chlorophyll pigment. The chloroplasts have a highly ordered array of membranes arranged in stalks called thylakoid membranes, as later acts as a solar panel with a large surface area and organize the pigment chlorophyll and carotenoids to absorb light energy. The chlorophyll is the principal pigment involved in the process. It is a large molecule and absorbs light maximally in the violet, blue and red region in visible spectrum and reflects green light, and thus leaf appear green in color. So, a large molecule and absorbs light maximally in the violet, blue and red region. Reflects green light and thus leaf appear green in color. Carotenoids, carotene and xanthophyll absorb light in the regions of the spectrum not absorbed by chlorophylls and transfer that energy to chlorophyll to be used in photosynthesis. Chlorophyll A, a special type of chlorophyll, is the main pigment that traps solar energy and converts it into chemical energy. Chlorophyll A is present in all autotrophic plants except photosynthetic bacteria. This chlorophyll A is called the essential photosynthetic pigment and acts as a reaction center. All other pigments such as chlorophyll B and carotenoids are collectively called accessory pigments, since they pass on the absorbed light energy to a chlorophyll A molecule to be utilized for photosynthesis. These pigments that is the reaction center chlorophyll A and the accessory pigments harvesting center are packed into functional clusters called photosystems. So photosynthesis involves two different processes: light dependent light reaction and light independent dark reactions. The structure of chloroplast is such that both these clusters of reactions take place at different sites in the same organelle. Light reactions. The light absorption part of photosynthesis is referred to as the light reaction. It relies on energy from the sun, so it occurs during the day in thylakoid membrane of chloroplasts. In light reaction, water split and oxygen released, but more importantly, it provides the chemical energy to fix CO2 into carbohydrate in the carbon reactions. It involves two photosystems, PS1 and PS2. Each photosystem contains different forms of chlorophyll A in their reaction centers. In PS1, each photosystem contains different forms of chlorophyll A in their reaction centers. In PS1, photosystem 1, chlorophyll A with maximum absorption at 700 nm, P700, and in PS2, chlorophyll A with peak absorption at 680 nm, P680, act as reaction centers. The primary function of the two photosystems which interact with each other is to trap the solar energy and convert it into the chemical energy. So, also called assimilatory power, ATP and NADPH2. After receiving light, PS2 absorbs light energy and passes it on to its reaction center, P680. When P680 absorbs light, it is excited and its electrons are transferred to an electron acceptor molecule, primary electron acceptor, pheophytin, and it itself comes to the ground state. However, by losing an electron, P680 is oxidized and splits water molecule to release O2. This light-dependent splitting of water is called photolysis. With the breakdown of water, electrons are generated, which are then passed on to the electron-deficient P680, which had transferred its electron earlier. Thus, the oxidized P680 region contains its lost electrons from water molecule. The reduced primary acceptor now donates electrons to the downstream components of the electron transport chain. The electrons are finally passed on to the reaction center P700 or PS1. During this process, energy is released and stored in the form of ATP. Photochemistry, light reactions, H2O, carbon fixation, Calvin-Benson cycle, photosystem 2, electron transport chain, photosystem 1, chloroplast O2. Similarly, PS1 also gets excited when it absorbs light, and P700 gets oxidized as it transfers its electron to another primary acceptor molecule. While the oxidized P700 draws its electrons from PS2, the reduced primary acceptor molecule of PS1 transfers its electrons via other electron carrier to NADP, nicotinamide adenine dinucleotide phosphate, to produce NADPH2, a strong reducing agent. Thus, we can see that there is a continuous flow of electrons from H2O molecule to PS2 to PS1 and finally to the NADP molecule, which is reduced to NADPH2. NADPH2 is then utilized in reduction of CO2 to carbohydrates in the biosynthetic pathway. Reduction of CO2 to carbohydrate also require ATP, which too are generated via electron transport chain. As the energy-rich electrons pass down the electron transport system, it releases energy which is sufficient to bind inorganic phosphate Pi with ADP to form ATP. This process is called photophosphorylation. Carbon or dark reactions. The carbon reactions occur in the stroma of the chloroplast and uses enzyme ribulose biphosphate carboxylase oxygenase. Rubisco is the most abundant protein in plants, therefore the major consumer of nitrogen. This is why when plants are deficient in nitrogen, they are not productive and turn yellow because they stop photosynthesizing. Both NADPH2 and ATP produced during light reaction are essential requirement for synthesis of carbohydrates. The series of reactions which catalyze the reduction of CO2 to carbohydrates, also called fixation of CO2, are independent of light but can continue in light as well as if products of light reaction are available. The carbon fixation reactions produce sugar in the leaves of plant, from where it is converted or exported to other tissues of the plant as source of both organic molecule and energy for growth and metabolism. The dark reaction above are found in more than 95% of the plants on Earth. There are two major pathways by which CO2 fixation take place is the C3 photosynthesis Calvin cycle, and in this cycle initially the atmospheric CO2 is accepted by a 5-carbon sugar ribulose biphosphate RUBP, resulting in the generation of two molecules of 3-carbon compound 3-phosphoglyceric acid PGA. This 3-carbon molecule is the first stable product of this pathway and hence the name C3 cycle is given. Formation of PGA is called carboxylation. This reaction is catalyzed by Rubisco enzyme. Further, PGA is reduced to 3-carbon carbohydrate called triose phosphate using NADPH2 and ATP from light reaction. Much of these molecules are then diverted from the C3 cycle and used for synthesis of other carbohydrates such as glucose and sucrose. To complete the cycle, the initial 5-carbon acceptor molecule RUBP is regenerated from the triose phosphate weights using ATP molecule. Thus, the C3 cycle continues to regenerate the CO2 acceptor or RUBP. Some example of C3 plants are given. wheat, rice and potato. These are C3 plants. The next is the C4 plants or C4 cycle or the Hatch and Slack pathway. C4 cycle. The C4 cycle mainly occurs in the plants growing under dry, hot environment. Such plants can photosynthesize even in the conditions of very low CO2 concentration and under partial closure of stomata. Therefore, these are efficient in growing at low water content, high temperature and high light intensity. Sugarcane and maize are some examples. The C4 plants minimize photorespiration, oxidation of RUBP in presence of O2 and water loss through a specialized cellular architecture, and the leaves light reaction occur in one cell type and the carbon reaction occur in the cells called bundle sheath cells or in direct contact with air. In C4 plants, the initial acceptor of CO2 is phosphoenol pyruvic acid or PEP, a 3-carbon compound. It combines with CO2 in presence of an enzyme phosphoenol pyruvate carboxylase PEP carboxylase and forms a C4 acid, oxaloacetic acid OAA. This fixation of CO2 occurs in the cytosol of the mesophyll cells of the leaf. OAA, oxaloacetic acid, I guess, is the first stable product of this cycle, which is a 4-carbon compound, and hence the name C4 pathway is given. OAA then travels from mesophyll cells to the chloroplasts of bundle sheath cells, where it releases the fixed CO2. C3 cycle operates within these cells and the CO2 immediately combines with RUBP in C3 cycle producing sugars. C4 species dominate very warm, high light environments, particularly tropical grasslands and savannas. Sorghum, corn and sugarcane. Some sort of cacti molds. CAM photosynthesis. Plants with crustacean acid metabolism. CAM photosynthesis pathway, such as cacti and all other succulents, minimize photorespiration and water loss by keeping their stomata completely closed during the day, so no water is lost. This also means they cannot take in CO2 during the day. CAM plants take in CO2 during the stomata at night and store it as a molecule called malate. Then during the day, with the sun shining and the stomata closed, Rubisco converts the malate to useful carbohydrate. Although this enables CAM plants to grow under extreme heat and dry conditions, they have low photosynthetic productivity and grow slowly. About 10% of the earth flora possess CAM photosynthesis succulents. The next, respiration. The organic matter produced in the process of photosynthesis is oxidized back to CO2 by all sources including combustion or by the respiration of plants, animals and microbes. Respiration is the exergonic biochemical process. Biochemical process in which glucose and oxygen combines to release CO2, water and energy. The released energy in form of ATP is utilized by the cells to perform the physiological activities of the organisms. Respiration may be aerobic with oxygen or anaerobic without oxygen. Aerobic respiration is reverse of photosynthesis. It is the process by which organic matter CH2O is decomposed back to CO2 and H2O with the release of energy. The equation of aerobic respiration is written as follows: C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus energy. All the higher plants and animals obtain their energy for maintenance and for formation of cellular material in this manner. During photosynthesis, solar energy is captured and stored in high energy forms in carbohydrates such as glucose C6H12O6. The latter is used by the plants or ingested by the heterotrophs. The energy contained in carbohydrates is released during respiration via glycolysis, and the Krebs cycle, carbon dioxide and water are also released. In virtually all ecosystems, photosynthetic autotrophs provide energy for the total system. Thus, the ultimate source of energy in the system is the sun. A sketch of the ecosystem respiration is illustrated. Anaerobic respiration occurs in saprophages such as bacteria, yeast, mold, protozoa, etc. The methane bacteria are example of obligate anaerobes which decompose organic compounds with the release of methane gas. Disulfovibrio and other variety of sulfate reducing bacteria are ecologically important example of anaerobic respiration because they produce SO4 to H2S gas in deep sediments and in anoxic water. The H2S gas rises to the surface where it can be oxidized by other organisms, for example, photosynthetic sulfur bacteria. Many bacteria such as Aerobacter are capable of both aerobic and anaerobic respiration, but the end product of these reactions is different and the amount of energy released is very less in anaerobic conditions. CO2 input in photosynthesis, CO2 released by plant respiration, CO2 released by animal respiration, CO2 released by soil respiration, CO2 released by microbial respiration during litter decomposition, CO2 released by respiration of root and soil fauna. Respiration at a cellular level. Respiration is the stepwise oxidation of complex organic molecules and release of energy as ATP for various cellular metabolic activities. It takes sugar either directly from photosynthesis or from breakdown of storage compounds like starch or lipids and uses its storage chemical energy to make energy currency ATP. It involves exchange of gases between the organisms and the external environment. The green as well as non-green plants obtain oxygen from their environment and return carbon dioxide and water vapors into it. This mere exchange of gases is known as external respiration or breathing in case of animals. That is a physical process. The biochemical process which occurs within cells and oxidize food to obtain energy is known as cellular respiration. The whole process of respiration may be divided into several steps. Various enzymes, biocatalysts catalyze this process. The process by which cells obtain energy from complex food molecules depend upon whether or not oxygen is present in their environment and utilized. The first step is called glycolysis, which literally means sugar split. This occurs in the cytoplasm of the cell and does not use oxygen and produces a small amount of ATP. Glycolysis also serves as the central primary metabolic pathway on which most other secondary metabolic pathways depend. This means that crucial plant biomolecules such as proteins, lipids, starch, cellulose, DNA, RNA, chlorophyll, other pigments, plant hormones and many others are intricately related with metabolic flux through glycolysis. The other part of respiration occurs in specialized organelles called mitochondria. This is where the bulk of the ATP is released in process requiring oxygen. Next, decomposition. Decomposition is a biological process of breakdown of complex organic materials, carbohydrates, proteins, amino acids, nucleic acids, etc. into the simpler one, CO2, H2 and other inorganic nutrients like nitrogen, phosphorus, sulfur, etc. It results from both biotic and abiotic processes. For example, forest fire release large quantities of CO2 and other gases into atmosphere and minerals into the soil. The grinding action of freezing and thawing and water flow break down the organic materials by reducing the particle size. However, by and large, heterotrophic microorganisms or decomposers or saprophages ultimately act on the dead bodies of plants and animals, and the release of nutrients is done by bacteria and fungi. The process bacteria and fungi obtain food for themselves. Decomposition therefore occurs through energy transformation within and between organisms. If it did not occur, all the nutrients will be locked in the dead bodies and no new life could be produced. Decomposition is a process of equivalent importance as photosynthesis and needs to be understood in its full detail. The Process of decomposition keeps the ecosystem going by release of nutrients back in the soil from where they are taken up by the produced cells. There are 4 stages of decomposition. Initial leaching. Leaching, the loss of soluble sugars and other compounds that are dissolved and carried away by water. Then fragmentation. Once first stage is leaching. Of particulate detritus by physical and biological action accompanied by the release of dissolved organic matter, then rapid production of humus. And release of additional soluble organics by saprotrophs. And next is the slower mineralization. Is the release of organically bound nutrients into inorganic forms for the use of plants with the help of ammonifying, nitrifying, and other microorganisms. So humus is dark brown or yellowish brown colloidal substance having nutrients in readily available forms to plants. Detritus, humic substances, and other organic matters undergoing decomposition are important for soil fertility. Initial leaching, the loss of soluble sugars and other compounds that are dissolved and carried away by water. Fragmentation of particular detritus by physical and biological action accompanied by the release of dissolved organic matter. Then production of humus, the release of additional soluble organics by saprotrophs. Mineralization is the release of organically bound chemicals and nutrients into inorganic forms available to plants and microbes. These are the 4 stages of what? Decomposition of organic matter. In aquatic muds, sediments, and in the rumen of ungulate herbivores, decomposition is carried out by the anaerobic bacteria, facultative as well as obligate anaerobes, through the process of anaerobic respiration or fermentation. In the decomposition process, the role of litter feeding organisms, mainly protozoans, microfauna, mites, mesofauna, nematodes, macrofauna, and megafauna comprises of snails, earthworms, millipedes, mollusks, and crabs, includes the fragmentation of larger materials into smaller particles. Their absence and overall decomposition may slow down the rate of process. During the process of litter decomposition, a large proportion of carbon is lost as respiration of decomposer organisms, and nutrients are released during mineralization. Decomposition is highly regulated by fire, moisture, temperature, and by the drivers of global climate change. Moist and unfairly temperature increases the rate of decomposition. Too less as well as too much soil moisture and temperature, severity of climate, and global climate change factors reduce the rate of decomposition. Herbivory refers to the ingestion of autotrophs such as plants, algae, and photosynthesizing bacteria by the heterotrophs. Herbivores can be of 2 types. Monophagous are organisms that exclusively eat one plant species, and the survival of these organisms is dependent on the survival of the primary food source. They are immune to the plant's defenses. For example, giant panda feeds on bamboo, and koala bears who feed on the eucalyptus leaves. Polyphagous feed on more than one type of plant. Frugivores eat only fruit. Folivores eat leaves. Nectarivores, nectar feeders. Granivores, grain feeder. Graminivores means grass eater. Palnivores, pollen eater. Palnivores, graminivores, grass eater, palnivores, pollen eater. Herbivory is a beneficial process for plants as well. Fruit seeds are dispersed over wide areas as the herbivores move. Those seed coatings are removed in the digestive tract of the herbivore, and its dung fertilizes the soil. providing an ideal environment for seed germination. Low-level herbivory can remove aging roots and leaves, allowing new growth of young roots and shoots. The new roots and shoots that grow provide better nutrients for absorption and reproduction. Herbivory may remove about 10% of net primary productivity. The percentage varies in different types of ecosystems. 2 to 3% in desert, shrub, scrub, 4 to 7% of forest, 10 to 15% for temperate grasslands, 30 to 60% for tropical grasslands and grasslands managed for cattle raising. Carnivory is the ingestion of animals or herbivores by other organisms. Consumers play an active role in the maintenance and regulation of energy flow through the ecosystem. So that's it. Thank you for listening.